So, thank you very much, and welcome back to Big Ideas at µScale. I'm really keen about introducing the next two people. In 2023, we had this opportunity where we had a chance to connect with the team out of the University of Waterloo, led by Veronica
Magdanz.
And in that time period, we've learned so much. And so I think they do some amazing things there. They've also been the ones that validated our new material that just came out, the Cyto-Clear. And I think they've got a story that is really worth sharing. So here we have Veronica and Noah Franco. My name is Hemdeep Patel. I am the co-founder of Creative CADworks, CADworks 3D, and ResinWorks 3D. My co-host is Robin. Come on,
and I'm Robin. Sorry, I didn't know I was meant to speak there. I thought you were still talking. You just cut out. Yeah, so I'm Robin. I'm part of the marketing team. Been with them for 3 years, and I didn't realize it was 2023 when you both started working. Waterloo and CADworks 3D started working together. It's been a while.
Yeah, it has been a while, and there have been a number of ups and downs, and I think we're going to go through that journey and the journey that Veronica and Noah have had, whether it's the last 2 years or even before that. So let's quickly introduce and bring on board Veronica and Noah. Hi guys, how are you?
Good, thanks.
Good, very good.
Happy to be here. Thanks for having me.
Yes, so 2 years ago, I had the opportunity of meeting up with Veronica and her team at the University of Waterloo. She and her team have been very important to the development of the current material, Cyto-Clear, and I am so happy to have a chance to bring her on board and introduce her to our listeners. Here she is, Veronica.
Yeah, thank you, Hemdeep. It's been great working with you guys. So just to introduce myself quickly, I am a biotechnologist. So I studied in Germany. That's where I grew up. And during my undergrad studies, I had the chance to go on exchange, and I came to University of Waterloo to study chemical engineering. And then as part of that exchange, I had to do a research internship, and I had the chance to go to Vancouver to work at UBC in a microfluidics lab. And that was my first time working on a microscale, and it absolutely hooked me. I really enjoyed working in microfluidics, but also just learning how to fabricate things on a microscale and push certain applications forward, especially in healthcare. And then after this internship, I returned to Germany to finish my degree. And after a few years working in different fields, I did my PhD in microrobotics. So that's when I joined a group in Dresden, Germany, that was one of the few groups worldwide developing these wireless nano or microscale devices for drug delivery on the microscale to revolutionize cancer treatment, but also other drug delivery to make it much more targeted and localized. Then after my PhD, I did a postdoc in Dresden as well on sperm biology. After that, went to Barcelona for 2 years to work at the Institute for Bioengineering of Catalonia. And that's when I first started doing more bioprinting and 3D printing. And then in 2022, I received the position here at the University of Waterloo as assistant professor. So I joined here in fall 2022. And as a way to start my network here in North America, I was looking for industry collaborations, because it's one very good way to fund research, but also just to build that network of industry and research collaborations. And at that time, I didn't know any companies in the area. And I was talking to my friend in Barcelona, and she said, I think we just bought a printer from a company in your area. And I said, oh, what's that company's name? And she passed me on the contact to Creative CADworks. I reached out to Hemdeep, and he wrote back right away, and that was sort of the start of our connection. And that went into a first MyTax project, and now we're on to the second one that's ramping up, and it's a bit more extensive. So that's sort of the beginning of the collaboration.
Wow. And so now when you were in BC, you said you were working with a team there. What team was it that you were working with, and what kind of work and research were they doing over there?
Yeah, that was the Carl Hansen lab at the time. I don't know if you're familiar with them. What we were trying to do was to run reverse transcription PCR on chip. So basically amplifying DNA and having everything on chip, from putting the blood cell there all the way to extracting the DNA or RNA, for antibody, from those antibody producing cells. Not much worked that time. I was there for those 3-4 months. I tried to do PCR, and I think it didn't work until like the very last week of it all. So despite being really painful and many trials, I just loved working in research. That environment was great. It was a young assistant professor at that time, Carl Hansen, and I think later he started a big company that had a big role in antibody production during COVID, actually, as a major therapy for COVID patients that were really suffering. So I believe he's not a professor at the university anymore, but is moved into industry completely. But he was just such an amazing supervisor that took me on, not knowing me at all, having this stranger German student coming undergrad and allowing me to work in his lab for the summer. That was really amazing. And at that time, we were not doing 3D printing. We were doing photolithography, right? So we had to go to the clean room, had to do all these multiple steps of making them, having the, what is it called? The masks. Photoresist. First making the mask, and then the photoresist coating, and all these multiple steps to get the chips in the end. So it was painful, but a good experience working in the clean room. But I think now we're like, 3D printing has just changed that field so much. So yeah.
Yeah. How long would you say it took to like make a chip in the clean room?
I'm trying to remember. I think it was at least one full day, like probably 9-hour full day, and then the next day you would do the lift-offs and then put them back together too. And it was one of those chips where you had multiple layers, because you had pump layers that have airflow going through, and then other layers that have fluids going through, and they had to be aligned like on the micrometer. So you'd sit under the microscope and try to align these different PDMS levels, layers. Yeah. So we can move beyond that now, I think.
Yes, we can.
Yeah.
And so now the second of our guests is Noah Franco. Noah, you jumped in right there before I even got a chance to introduce you. So here you are. So why don't you give us a quick history about yourself and the stuff that you've been doing up until now. Go ahead.
Yeah, of course. Thanks for having me on. So I'm a master's student at the University of Waterloo, doing systems engineering at the Magdanz lab. And I guess I come from a more non-traditional background coming into engineering, because I did my undergrad in Waterloo, and I was in the science and business program. So it was kind of a mix of biotechnology and business internships, but I always seemed to like drift towards the research. And so while I was at Waterloo during my undergrad, I got to kind of delve into all types of R&D. My first co-op was in Toronto, working on clinical trials of the Canadian-made COVID vaccines, and testing the first-in-human trials for like Medicago and stuff. And that kind of got me interested in med school and research and stuff. and in the right direction, I went to Ottawa then, and that's where I looked at virus-based COVID vaccines, and also cancer vaccines. So we got to do a bunch of cool work there. Got my first publication from that internship. And then from there, I wanted to go a little bit more into engineering just to like see what it's about. And even though I wasn't in an engineering program, I wanted to get the best of both worlds, to kind of experience everything in one. And so I worked at a really cool company called Tersa Earth. And what they do is they're a bioremediation company. So they're basically pioneering a process to take like acidic rock drainage from mining waste and process it to extract any leftover heavy metals. So like a higher yield on the waste, and then also purify that waste. And so I was the like genetic engineer there. And so we looked at modifying different types of bacteria to be used in microbial fuel cells for the processing of the waste. And then my last one was a little bit more business based. I did business and R&D for a company called Alora that was in Waterloo. It was Velocity at the time. But then, yeah, from there, when I was finishing my undergrad, I joined Veronica's microrobotics lab, and I was super interested in how I could spend my master's being able to use microrobotics to kind of tackle a problem in healthcare, which has always been really interesting to me. And yeah, so it was really cool, and been getting more and more into 3D printing, first through design teams and then through working with Creative CADWorks to make these really cool resins.
So you mentioned Veronica's lab, the Magdanz lab. When did that actually start up? And you mentioned, was it micro robotics? What are kind of like the other research interests that goes on in the lab?
Yeah, I think Veronica can definitely touch on this for sure. We do have some cool projects.
Yeah, and then you can also describe sort of the projects you're working on. Yeah, so the Magdanz lab, like when I look at the website, I've had it for probably 6 years now, so way before I came to Waterloo. But the projects have evolved a bit. So first I worked mostly on sperm diagnostics and sperm-based microrobots, which might sound a little strange, but it sort of goes way back to when I did my PhD on sperm-driven microrobots for applications in the reproductive tract. So that's still one of the lines we do, but we've now expanded to also working with synthetic wireless microrobots, or let's say really man-made fabricated small-scale robots for so one stream is for removal of occlusions in the body. So that could be removing blood clots, removing kidney stones. Noah will also talk about another application he's exploring, as well as drug delivery or cell delivery, all as therapeutic applications. We have some work also still on sperm diagnostics with new techniques. And then we have one stream that's a bit more on 3D printing, bioprinting, especially to create sophisticated in vitro organ models. So the idea is that we can eventually really print a little organ that combines cells as well as a bioscaffold to use that as an in vitro model for drug testing or other microbiologic testing. Those are sort of the main topics we're working on at the moment.
Yeah, so I can jump in and talk about my thesis again because it's lots in there. But so started my thesis about 8 months ago now, in September. And what we're working on is developing a non-surgical treatment pathway to treat gallstone disease using microrobotics. And what we want to do is be able to develop robots that can be loaded with drugs and non-surgically guided into the gallbladder, and then be able to dissolve and break down the gallstones. And just a bit of a background on gallstone disease. It's actually quite common. 1 in 3 women will be impacted by it at some point in their lives, and 1 in 5 men will be impacted by it as well. It's more prevalent in women. And the problem with gallstone disease is that you don't know you have it until it's too late. You really only get symptoms from gallstone disease when these stones become big enough to block your bile duct. And when it does become too late, the only solution doctors really have, and that they've reverted to for the past 30 years, is to surgically remove the gallbladder. And that's called a cholecystectomy. It's one of the most common surgeries done in North America. And so
actually, I have a friend who had it last year, so
my mom had it, and she was no, sorry. It's so funny when you said that, because my mom had it a few years back, and I kid you not, for nearly, I think, 4 months, she would just go, "Oh, I think I've got this ache in my stomach," for 4 solid months. And when she finally went under, the doctor came out and said, your mom is made out of a ton of bricks. She should have been withering in pain. It had gotten so atrophied. And and then when that happened, I found a lot of people have had that happen, and then the onset of pain is almost immediate at that point and excruciating.
Yeah.
So,
Yeah.
wow.
Yeah. Robin, did you want to say anything?
Oh no, it's just like I wish these micro robots were already done, because I had a friend, she also had it just last year, and this was around the time that she was giving birth as well. So
Oh wow!
you could kind of imagine,
Crazy.
the pain that she must have been going through.
Yeah, from the people I've talked to, the consensus is that people always want like another option. Because there are like consequences to having it removed. I mean, like you have to change your diet as well. Like Hemdeep, did you notice that? Any like lifestyle and diet changes that happened?
No, my mother is still just going by her. It has not changed a bit, I'm sure. Yeah, her rules, her life, and I don't believe. I didn't even know. I'm sure that the doctor probably gave her a list of things to do and not do, and I think it sort of got tossed as soon as she stepped out of the hospital. I don't think she had a concern in the world. She was like, "Okay, great, I can start traveling again." That's all she really is her big passion in life. I had one question, and the one question is that for both of you, in fact, it seems like you went through all of these points in your life of joining different team, whether new labs, new projects, and that involves or requires a lot of learning because it's a brand new topic that you're learning. How much of that was the ability to absorb new topics, or was there a unique precursor that you had that allowed you to go from topic to topic to topic, ever changing, and yet be able to sort of pull old experiences to a new topic and still sort of get on with it and really sort of jump ahead and take the topic, whatever new subject you had forward?
Yeah. I mean, from my perspective, going into all these different co-ops, I don't think I had something special other than just like a passion and a will to want to learn new things, and that drives you to learn more than you thought you came for. And I think that's what I tried to do during each of my internships, was just pick something that I wasn't comfortable with, pick something that I wanted to learn rather than something I already had the skill set for, to kind of make myself uncomfortable and to be able to learn new things. But yeah, that's my take on it.
How about you, Veronica?
Yeah, I mean, we constantly learn new things, and that's what I love about research. That, for example, I didn't know anything about gallstone disease going into this topic, and basically was throwing it at Noah and said, "Do you want to look into that and see if that makes sense. I think what I would hate the most, having a job where I would have to do the same thing over and over again, or even having to do the same thing today that I did yesterday. So every day is completely new, trying out new things, learning. It's constant learning, and it's really satisfying, right? It's really, really great being able to explore.
And then how does like obviously when you're learning a brand new topic, or even sort of creating a brand new topic out of nothing, there's this level of expectation of success, but then there's an overwhelming expectation that none of it's going to work. How do you guys manage that? Obviously sometimes, in our case, there have been, you invest months and years into a topic, and then it all is all for naught. How do you manage those?
Yeah, I think for me, the strategy has always been that I never just focus on one thing. So I usually have 2, 3, 4, maybe 10 different things going on. And that probably most of them won't work out the way I hope. But then we live off those little successes in that one project that does work out. And it's so exciting when you see things happening in the lab, or trying out experiments that you've never tried out and nobody has ever tried out, and you tried and it works. It's just so amazing. I think we live off those moments. And then on the other hand, we do draw experience also from different projects. So despite having so many different projects, we have a common theme, especially across the microrobot research, where it's all about making therapies more targeted and more actively delivered. So that is true for like gallstone treatment, but also blood clot removal, kidney stone removal, even antibiotics delivery. Basically, like looking at the conventional treatment, or also cancer treatment, where you have drugs going into your whole body because you take them either by IV or by oral medication, and it affects your whole body. And the idea is to just make these therapies much more targeted and more successful, and get rid of all those side effects. So that's one of the, I think, common themes for the microbots. And the other important thing for us is that we go into completely new fields, and that's the exciting part. Yeah. And we're not competing with 20 other groups around the world that do the same stuff. Noah's probably the only one working on what he's working on. And so it's also a bit easier to be successful, because whatever you get out is a result. It's something you can share and is worth sharing. So yeah, I think that way it's also exciting.
Yeah, for sure.
Considering that you're at the starting phase of your life in academia, how do you sort of manage expectations and the, this level of failure or expected failure?
Yeah, it's definitely scary, especially when you come up with something and you're really passionate about it, and that's the thing that you want to see work. But yeah, like, you have to balance that with the fact that it's very entrepreneurial, and when you're making a company and it's successful, that's the best thing ever. But they almost always fail. So I try and balance my expectations with the fact that it's okay if things don't work, but at the very minimum, we can at least make like a framework for future researchers to build off of it. Because at the very minimum, I want to be able to introduce something new and unique that can be able to benefit the field, right? So it's definitely scary, but I mean, like, it's the little steps that you have to like use as milestones to kind of keep yourself going.
And so, what do you find is what are the characteristics then? Would you say that allow you to do that? Because so, for example, in business, obviously, is you're either you evaluate risk in a particular way. You either are adverse to risk, or you're okay with it. what is it that what are the traits in the field of academia like that really make it a successful researcher, and one that sort of may want to just continue doing the exact same thing day in day out?
Yeah, like you definitely have to have a bit of an entrepreneurial mindset to want to go and pursue something new. And I think that is definitely important if you're pursuing a project, but you don't have to. There's people who don't. There's people who just do their master's and their PhD and go into work. But I mean, like, having a project and having an entrepreneurial mindset will let you take it that step further, to the point where it's more than just a degree or a program. It's actually something you can call your own and something that could have an impact.
Yeah, I think it's also that curiosity that you have to really embrace and live out. And that's where I usually see students be successful too, when they are really curious, they really own the topic and just go at it. Like, just be super proactive. Just go to the lab, try out things. Don't read for 3 weeks and see, oh, this could go wrong and this could go wrong. No, just go and try it out, and learn on your own. That's where I see like a lot of success coming out, and of course failures too. But failures are never, it's never the mistake of a person. It's a failed experiment. It has nothing to do with me or with Noah failing. It's not our failure. It's just a result that we should consider and take serious. But I think it's also important to distance ourselves from the results in a way that this is not my self-worth that I'm dealing with. But the other thing I want to just mention that may be a bit different to what Hemdeep mentioned of, when you do your risk analysis and so on, we have the privilege, and I really see that as a privilege, to work in research, that I'm still going to have my job tomorrow if my experiment fails today, right? Whereas in a company, that's not a given. If you're not selling, right? If things are not going well, this really affects your company and your people. For me and Noah, this is, and like I said, this is a privilege, being able to work off of research funds, to have that freedom and curiosity and follow that. I think it's really an honor, and that's sort of that the way I see it for the research.
Yeah, it's good you brought that up. But then on the flip side, Hemdeep, what's your thought on failure within the industry side of research?
You know what, industry, the number of failures that I've had, epic failures. We're talking failures that were setbacks of like, a 4 or 5 year setback. That type of failure, we've done it, and through it all, it's been myself, my brother have co-founded things, and the failures that we see and that we've done, in retrospect, now that we're in our 50s and we look back, we can actually we can map it out where our shortcomings were, where we were, navel gazing, and we were not planning out, really strategizing for epic failure, and we were very thick-headed, and all of our attributes that are really wrong, we realized that we had to modify that. And so over the last, let's say, 5 or 6 years, we've gotten a lot better with really understanding what we have, what we don't have, where our gaps are. The one thing that I tell our team every day is the fact, well, maybe not every day, but it used to be every day. Now it's like once a week or once a month, we have this moment where we just sit down and we speak of what is our blind spots. And it's a moment of reflecting on, yes, we may be doing X, Y, and Z well, but there is a spot where we just don't see, there are mistakes that are sort of compounding on each other. And eventually, when you find it, it may be too late. So can we identify that? And so that seems to be the one big thing but then in terms of risk, we're very, very, we're poor in evaluating risk. We seem to run headfirst. And I think even during our interaction for the last 2 years, there have been moments where I'm sure the two of you, and even before this with Lydia, probably scratched their head and just said, oh God, these these guys are just, what are they doing? Why are they doing it this way? This is not the way that you're supposed to do things. And it's so, it's almost our fabric of the way that we function. It's, I do believe it's a fault, but it's, we haven't killed ourselves. We have we've nearly burnt down my mom's home. We nearly burnt down our office that we used to be at in the city itself. And when I mean burnt it down, we almost burnt it down. There was a fire. There was a alcohol.
From a 3D printer?
We had taken. So there was a material that my brother was sort of manufacturing, and he goes, "Look, you know what? This thing has got amazing components, and you know how we have to use IPA to clean our models." So we were on this large workbench, and right beside a wall, and the workbench itself had IPA sort of, there's a very thin layer of IPA on the workbench. We didn't notice it. But my brother wanted to sort of take a model and light it just to sort of showcase. And he didn't realize it, but a drop of it fell on the bench. And we didn't notice anything until this blue flame just right up the wall. And we're like, oh, no.
Oh, wow.
Oh, my God.
Was this at your mother's house, or was this the one in the office?
No. Oh, no, this was at the office. My mother's house, we burnt her kitchen. When I was 13 and my brother was 9, we burnt her kitchen. She was away at work. I kid you not, we burnt the ceiling, the stove hood, and then, like a typical 13-year-old and 9-year-old, we proceeded to invite all of our friends with paint and brush, and we repainted my mom's ceiling. Yes.
Can stress cause gallbladder? Because gallbladder stones? Because I think we know where it came from.
It was the best thing. I have never. My mom came home that afternoon, so we did this all in the afternoon. So we burnt the kitchen at around noon. By 3:00, we had the place painted, and it was it was in the summer, and my mom's coming home, and the house has paint fumes everywhere. We've opened all the windows. We've got fans going. My mom walks up the street and says, "Why do you guys, what happened? Why are all the doors open?" We go, "Oh, don't worry about it, Mom. We were just airing the place out." And she goes, "Why are you airing it out?" We go, "Well, we thought that we could paint it up just to liven up we decided to paint this area." She goes, "Why would you do that?" And so it didn't dawn on her that her sons would have decided to pull this. She goes upstairs to her bedroom and she notices that there is smoked soot that had fallen on her bed. That's how much we lit the place up. And she came down and she just had it. She chased us down. It was epic. And that's the starting of a great relationship, right? This is how you find your co-founders.
Nice.
Yeah. So that's
funny.
the story of my life.
So you've been doing this business for a while now, or how many years have you been working with your brother?
Oh my God. I think forever and a day. As soon as I joined my dad's business when I was 23. He then joined our business when I was 27. So 27. So we're now closing in on 30 years together.
Oh, amazing.
Yeah.
So did you ever have, if I may ask, did you ever have a point where, because you described you had also lots of downhill times or failures. Did you ever have a moment where you were going to fold it and say, that's it?
I don't know about folded. I think we really were wired in this way of like, okay, so what do we do now? It actually as a team, what do we do now?
Nice.
So we've had major deflection points. The one that we had in 2009 is when my dad had passed away. At that point, that was a really big change in where we sort of reorganized ourselves. And it was in 2009 that we took up 3D printing. We self-taught ourselves CAD design. So we now, at that time, it was a really big reset in our life. But when we looked at it as, okay, what do we do now?
Yeah.
It certainly wasn't. I don't recall any time ever sort of saying, oh, I don't know, maybe I need to sort of let's part ways. I don't know if that ever crossed my mind. We've had many moments where we didn't connect in terms of in strategy, but then we really started designing our businesses where, okay, you're really good at this, like in this business, he is our CTO, so he manages whether it's all the technology side, the photopolymer manufacturing, formulation, and all of that is his, and I do all of the business side, and I have a very good understanding of what happens on the technology side, but that's not the thing that wakes me up in the morning. The thing that wakes me up in the morning is trying to distill complicated processes or technologies into a format that really is can be understood by a wide range of people, and that also excites them. And when I connected with your team 2 years ago, it was purely on this. Oh wow, this might be interesting. This is something that we'd done before. We had done one with a collaboration with the U of T in 2018. I did a collaboration with the Majid team from UTS Sydney in 2018, 2019. And those ones we really enjoyed those collaborations. And it's so funny, when our collaboration happened, I had approached a couple of teams prior to that, and I think they sort of looked at us, and they were like, "Oh, what do you guys do?" And we explained what we wanted to do, but there wasn't a keen curiosity or interest. And so we thought, okay, well, maybe it's not the right time. And then you guys came along, and it was a really good opportunity and a good relationship that I think we've built over the last couple of years.
Yeah. So how did that actually happen? Like, how did you find out about CADWorks 3D? Like, were you searching for something?
So I started as assistant professor here in Waterloo in fall 2022, and it was a big change for me. I came from Barcelona and had done my postdoc there at the Catalonian Institute for Bioengineering. So had to start building my network in North America. And as you probably know, one of the big funding opportunities in Canada is in collaboration with industry. And there are programs like MyTax that really support that. So I was looking to build a network with industry, with companies that are also interested in doing research together. And there was a call from MyTax that was going to support collaborations in the biotech field between academic research and industry. So I was looking for local companies, but really I didn't know at all the area. And I was talking to my colleague in Barcelona, and she said, I think we just bought a printer from a company that's really close by. And I said, really, what's their name? And she said, yeah, this Creative CADWorks, and passed me on the contact. And I wrote an email to Hemdeep, and he wrote back right away and said, sure, let's talk. And I was really amazed about the interest in doing collaboration with us, because it's not a given. Often industry runs differently, has different interests, but there were, I think when we were brainstorming, there were a few topics right away that we could see there could be some common interest. And so, yeah, we started going into our first MyTax collaboration, and now we're on to our second one and are expanding. So it's been really great.
And Hemdeep, so when the collaboration first started, actually, how far along was the Cyto-Clear in development?
Oh, it was on the shelf at that point. It was at that time, it would have been on a shelf for about, I think, 3 years. We had we had it running. We were very happy with it, but there was some significant shortfalls that we noticed, and we wanted to get it validated. There were a lot of steps that were still left to do, and we weren't very it really wasn't on our radar at all. There was a sort of, oh, we should get to it, but I think when that email came in fact, it's so strange. Veronica says it was a conversation that she had already had with Barcelona. I had got an email from a student in the UK that came to us and said, "Hey, you know what? I'd like to do. I was just wondering. I'm going to be at Waterloo. Would I be able to intern with you as the industry with Waterloo?" And so that's where our connection happened. And then at that point, it sort of dawned on us that, oh, you know what? What about that clear material that we've been sort of kicking around for the last 3 years? Is that something that we can sort of bring to the table and see if we can sort of push it through and figure out if we can get it validated, really understand what do we have on hand. And that was the starting point for us. We were very excited when that email came in, that there was an opportunity, because up until that time, as I said, we had a couple of conversations with some teams across Canada, and there wasn't a lot of sort of reciprocal curiosity about what we were doing. And so it was like, okay, all right, maybe we might have to sort of wait a bit longer for that ideal partnership at that point.
And then were there like other iterations, like after it got picked up and you put it into Waterloo's hands? Yeah. Were there like a lot of iterations that followed that, or yes. What did that look like?
Yeah, I think Veronica can speak to the frustration. I think that is that's the big difference between academia and industry is there's a full-on mindset when an industry person comes along, and that is and what the mindset of academia looks like, and 9 times out of 10 they don't really match. And in this one, I don't think we butted heads, but you can see that our iteration cycle didn't match up with what Veronica and her team
were there were some changes in the formulations that required us to basically go back to step 0. Yeah. But it's not like we're not used to those kind of cases. We do things over and over again, and like until we make them work. So I think it's going quite well. I think we're also finding a good rhythm of working together, and we're always happy, when we get a package from Creative Catworks. Oh, another resin to test. So I think it keeps us going too.
Yeah. Noah, what has your experience been with us? Don't hold back.
Yeah, that's fair. I mean, we went through a lot of iterations in September, October, like early on, just like every week, banging them out. But yeah, everything went well, so there was nothing really like frustrating.
It was just If I may say, we also have to acknowledge that there was an exponential improvement in the resin, right? When we started off, we sent the first one. I thought, there's no way we're going to make this biocompatible. Like, it killed the cells immediately. And then the next version already was way better. And then I think where we're now, it's just amazing. So that was really good.
Yeah, I think I need to give credit to Lydia. I think because she had come up she was with us on the first phase, and I think that was where she carried a ton of the frustration. And and that's the one thing. And this is more of a sidebar. I think Veronica managed to find. And I think the team members that she's bringing on board have this capability of going through the rigors of research, and that the expectation of success isn't sort of set in stone, and that the developing new skills is something that is held in really high regard. The ability to go through the steps and go through it over and over again. Lydia was was amazing to sort of pull that one through. And despite it not working out at the end, she really sort of persevered to making sure that at least the strategy and the testing, the protocol that she developed were things that became a really strong baseline that we can now, we were able to build on in the second phase.
Absolutely.
100%.
Yeah, that's the other thing also that I think is sort of the core of those MyTax collaborations. That's really meant to be a training platform for young researchers, for young scientists. So that's why they don't even ask for results, right? They ask for, okay, what did the student learn? What is the training we have provided? And so that's also always good to keep in mind, that it's not just the results, but it's the training and the establishment of protocols and so on.
More into that. How about we actually start talking about what really went into the beta testing of the material? I think you did tests on 5 different cell lines, or is that?
Yeah, we did 5, and so moving for the past 8 months, we've been pretty much testing the cytotoxicity and biocompatibility of the Cyto-Clear and moving through the iterations. We worked on a bunch of cell lines, and we kind of wanted to get a broad spectrum attribute to the resin where, it could be used for any kind of downstream use, any kind of cell line. So we kind of tested, we split it up into 2 different types of cells and applications. That was adherent and suspension cells. So the suspension cells that we tested with the material were sperm cells, and we tested monocytes, so blood cells pretty much. And then for the adherent cells, we looked at a uterine cell line, mesa cells. We looked at an endothelial line, so vascular cells. And we looked at keratinocytes, so skin cells. And we basically came up with 2 types of assays to test the cells based on their applications. For the suspension cells, the free-flowing ones, we came up with an on-chip assay to test the cytotoxicity, where we would take the cells and we would print our own microfluidic chips, and we would incubate the cells inside the channels of the chip, and then we would do a live dead stain inside of the chip and be able to image right on the chip, which is really cool. And that's how we tested the suspension cells. When we were testing the adherent cells, what we did was take their regular 24-well culturing plates, and we printed discs that would be able to slot right into the culturing plates, and then tried to get the cells to grow around or on the discs, and then measured their viability with not just a live dead stain, but an XDT assay as well to look at the health of the cells. And they reacted well to everything, which was really cool. And moving from cell line to cell line, we were able to touch on each cell line faster than the last one, just because we knew what the baseline was, and we weren't worried about anything happening to the cells at the end.
For the free flow cells, because it was on chip, right? So I guess you placed it under the microscope. Was there actually any difficulties viewing what was going on with the cells? Was the resin kind of inhibiting the ability to see what was going on inside the chip?
No, by the end, we were able to print out nice rectangular channels that didn't impede with the laser wavelengths from the microscope that live and dead stains need to be seen under the microscope. So it didn't inhibit the fluorescence. And on our own end, we had to work out some printing parameters and post-processing to get the chips really clear. But by the end, we were able to do the assay right on the chip and see the cells in their clusters and free flowing as blood and sperm would, which is really cool.
Yeah, I think that was also one of the really nice outcomes, nice sort of side result, that we decided to do the assays on chip, and for bright field as well as the different fluorescent channels, we were able to analyze the cells really well on chip. So that was also a nice sort of validation of the material.
I remember when I think I didn't realize that you'd been doing this until I was in India and you had called me and we had a meeting and you started showing me images. And I think at that time it dawned on me, because as you were showing me the images, you had sort of just as a matter of fact said, "Oh, by the way, these ones were imaged right on chip." And I remember when you said that, I was like, wait, hold on. What did you just do there? And I think that was, I think that almost validated the entire setup that we had, where the big gap between industry and academia is that we can build a better mousetrap, but sometimes it's academia that actually shows you exactly what the applications can be and really what secondary applications. So in our case, we were just thinking of building out a very nicely transparent biocompatible material, and that really was our basic need. But this team actually put it all together and sort of took it one big step further and validated across broad spectrum cell lines, and then be able to do validation on chip was something that we never even thought was going to be possible with this material.
Yeah, I think it was a good mix between academia and industry, taking the strengths of each, where like industry is really fast moving and can prototype out what you want to see very quickly, whereas academia is more in depth and we want to see everything before we move on. But yeah, that was one of the cool results of that, and we were able to take a look and apply the material to what its downstream use might be.
No, I was going to say that I think it has the potential to be used in many research areas. The chip, but also the resin, but also the technology itself, and for research on the one side, but also for industry. So we can see potentially these chips to be printed for point of care devices or even for home diagnostics. When I'm thinking of some of the sperm tests that are important to look at infertility, but also for point of care in hospitals, to print chips for blood cell analysis and so on. But then, like you say, also for research where there's no limit, like you can use this material for so many different areas. So our idea was not to demonstrate all these different applications, but just sort of show the baseline and say, look, this material might really work for a broad range of cells.
Yeah. I mean, I'm glad you brought that up. There's definitely a ton of uses, but even for like POC devices, point of care devices, it would be really cool to have a chip that can take very simple like blood cells and run the tests that they're running that they would have to spend 2 hours in a lab doing, like the monocyte monolayer assay. They do this in the hospital when someone needs a red blood cell transfusion.
Okay.
And it's basically a lab test to make sure the antibodies of the recipient aren't going to react to the donor's blood. And so there's so many applications for this material on POC devices and like lots of other strenuous lab tasks.
In terms of the testing that you did with this material, what additional tests would you have wished you would have been able to do? Like, I know that the 24 well, you had done discs instead, you had done discs that you would have laid down in each of the wells, would you have wanted to do additional tests, or what would that have? What kind of information would that have imparted as well?
Yeah, there's definitely a ton of directions we can go in the future. Some other things that we want to look deeper into is maybe cell adhesion, looking at like simple surface modifications to the material, and then going and testing cell viability and health on the adherent cells to see if they'll go and grow on the 3D printed material as well. Um, Monocyte differentiation. Inflammation markers for the blood cells. There's a lot of directions we could go.
Mm-hmm. Yeah, especially if we envision a material as scaffolds, or for any application that would be for longer term, let's say more than a couple of days. There's definitely more tests to be done that what Noah mentioned, that enhance the cell adhesion and differentiation and proliferation. So that's something that would have to be developed cell specific and application specific.
All right. Well, I do have a couple more questions more about the 3D printing side.
Yeah.
Did you get any print settings from us when you first got the materials, or were you going in completely blind regarding that?
I think that was a lot on during Lydia's time.
Okay.
So we got some settings, and then we had to tweak them a bit. And then obviously with every new resin batch, it sort of has to be adjusted a little bit. But I think, Noah, now it's pretty straightforward, I guess.
Yeah, it's ironed out. I haven't changed it since the end of last term. So like, yeah, Lydia ironed out the basics for sure. And then as we moved through the iterations, it was just small tweaks to the print settings. But yeah.
Okay. So like, how long would you say it takes you to because you had different iterations, right? How long did it take for you to validate the new print settings between the iterations?
Pretty quickly, like couple prints, so less than a week per iteration maybe. The stuff we print isn't too big or anything, so like the chips will usually print less than an hour. The discs that we print, I was just printing discs at the time when we were validating it, and those would print in like 10 minutes. So it was very quick, like iteration to see what worked and what didn't.
And was also Lydia who kind of made the post-processing procedures, or was that more of like your tasks?
Yeah, I remember she tried out quite a few things, and I also remember that particularly one of the washing steps after post-processing really improved the biocompatibility. So I think we do a water soak and UV exposure.
Yeah, so you're right. Yeah,
that's something she tried out, and I think that's really helped.
Yeah, she found that she looked at viability for different types of post-processing cures specifically. So yeah, she did find that curing in DI water for like a super long time does increase biocompatibility quite a bit.
So I think that is the end, or that concludes this conversation. What do you think, Robin? Is there anything else that you wanted to add?
No, I think we covered all the bases, really.
Yeah. Okay.
Great.
So thank you very much, Noah and Veronica, for joining us today. I know that I was very happy with doing this. I'm glad that our audience and all the listeners out there at least got to know exactly the team behind a lot of the work that we do on our side. And I'm so happy that everyone gets to hear about what you do and the type of other work that you're doing as well. We will add links to Veronica's team and any additional links to some of the things that we've spoken about in the show notes. And so that concludes this episode at Big Ideas at µScale. Thank you very much for listening, and we'll see you on the other side.
Thank you.
Thank you for having us on.